everyone. I'm joining here from Switzerland, where Studica has its headquarters and is advancing our platform-driven effort to develop precision medicine for subgroups of patients with neurodevelopmental disorders, with the first application in Autism Spectrum Disorder. So I'm the CEO and founder of Studica. I founded Studica 7 years ago on the vision that the neurodevelopmental disorder community was not benefiting from translation from basic science into clinical development, and that we needed to address through problem-solving approaches the heterogeneity, the intrinsic heterogeneity within the neurodevelopmental disorder and ASD neurological entities to be able to have an adequate biologically founded starting point for drug development. Today, Studica has matured its pipeline. We have a team of 25 people between Geneva, Switzerland, and Barcelona, with our team of computational biologists, data scientists, chemoinformaticians, and human geneticists based around the supercomputing center in Barcelona, who are developing and scaling, as we get in more clinical data, our precision medicine platform dedicated to precision medicine neurodevelopmental disorders. Studica's strategy rather than business model, because today is about strategy for precision medicine and patient centricity, relies around the DeepHi platform, and the core data is integrated into our platform. Our internal drug development pipelines, which are internal pipelines that are developed on the basis of the subgroups of patients that are characterized and identified in prospective observational studies. Collaborative pipelines, where we collaborate with third-party biotechs and pharma, leveraging on our platform to either rescue or advance in precision subgroups the novel treatments for patients with neurodevelopmental disorders, and obviously our proprietary clinical and biomarker database. So our platform now holds subclinical validation, and it was really structured to be able to identify biologically enriched subgroups of patients with NDDs that we would then validate, hypothesis-driven, then validated in prospective clinical studies. So we have done this for 2 subgroups of patients, ASD, so-called ASD phenotype 1 and phenotype 2, and we have also shown our capacity to revisit previously failed clinical trials, collaborating with investigators such as Elizabeth Berry-Kravis, Yipo Bernivia, other investigators in the ASD field, and revisiting failed clinical trials, blindly recalling with specificity, high specificity and sensitivity responder patients in these previously failed clinical trials. So today, in terms of tangible assets that we're bringing forward for the ASD community, we have our lead clinical pipeline, which is lead pipeline being STP1, specifically dedicated to benefit ASD phenotype 1 subgroup, which we will detail further down in the presentation. STP1 is now phase 2 ready, so we are looking to enter phase 2 clinical trials in mid-2023. STP1 is a composition-protected fixed-dose combination of a PDE3/4 inhibitor and KCC1 antagonist. It's specifically tailored to this phenotype 1 population. STP2 is a small molecule. It's actually a synthetic stabilized, cyclodextrin stabilized form of sulforaphane that we recently in-licensed for a second subpopulation of patients with ASD that we validated in a prospective clinical setting. And we will also be announcing in the coming 3 weeks the in-licensing of a phase 3 ready asset, which we, from a large Swiss biopharmaceutical, which we will be positioning in a specific subpopulation of patients with ASD and beyond ASD in other neuropsychiatric disorders. So obviously, today is not a place to talk about business. If you want to be able to advance treatments for the neurodevelopmental disorder population, you have to operate within the biotech economic ecosystem, and you need to protect your development. So we have a strong IP strategy around this precision medicine development strategy. We were recently granted a European grant, which is the equivalent of an NIH grant here in the U.S. For our computational biology drug computational biology drug repositioning efforts, leading this European program. So why have we developed the DeepI platform in a problem-solving approach? Well, because we, as a group of founders in the company, have very strong ties with the neurodevelopmental disorder community, myself personally, but also our head of discovery, and we saw failure after failure in the last 15 years, and we sort of reversed the problem, saying why are we failing? What are the intrinsic reasons that can explain why the ESC field is not going forward alongside oncology when we are leveraging on the same type of progress? And one of the commonalities between oncology and immunology field and the neurodevelopmental disorder field is that we have an intrinsic patient heterogeneity among and within neurodevelopmental disorders, meaning that within autism, we consider autism like concentric circles. So if you see one patient with autism from a biological standpoint, this patient would be closer to another patient who holds a DSM-5 clinical diagnosis of schizophrenia than to a third patient with autism. And so this patient heterogeneity really needs to be addressed, and I believe that in the last 7 to 8 years, maybe 5 to 7 years, we are now seeing tools that allow to address heterogeneity in complex disease settings. So the other reason for failure, and I think it's a very fundamental reason, is actually poor consideration for how nosology, so DSM-5 definition of autism, but also other neuropsychiatric disorders, is constraining drug development. Because if we're asking the wrong question, if we're saying, okay, how can we treat autism, but that autism does not have fundamental biological construct, then we are not equipping ourselves to have a starting point for drug development. So reliance on these behaviorally defined disease entities has been a hurdle for drug development. Obviously, from a regulatory standpoint, you have to go somewhere. So there are ways, and it's a separate discussion, that this can be addressed from a regulatory standpoint. An additional barrier to progress in the field appears to be hyper-focus on genetic data as the only valid level of data versus further integration of genomics and biosystems. And we really need to see genetic information as one layer of information, but the whole picture is more complex in the sense that patients who have, most patients with autism have additive risk variance, almost an individual combination thereof that go downstream at the pathway level and can converge in subgroups. So we have to be able to address the patient heterogeneity through multiple layers of data, and this has not been done. People have a tendency to hyper-focus on specific layers of data rather than multiple data or integration. And then this results in the impossibility to identify consistent biomarkers. And I think that the fundamental word here is consistent. It's not that the biomarkers are not there. We are talking about perturbations that have system expression. So we're not only talking this is a clear place where everyone understands this about CNS manifestations, but about systemic manifestations, because genes and molecular pathways that are expressed in the CNS are also expressed in other organs and diseases. And so there are biomarkers, but if you're asking the wrong question, if your question is, are there biomarkers for autism? No, there are no biomarkers for autism. There are biomarkers in subgroups of patients with autism, and that is the starting point to be able to advance drug development. And obviously the usual suspects in inadequate study design, endpoint sensitivity, etc. But what we really believe is that if you get sufficient biological signal and convergence, and you start addressing biologically convergent populations, then you will start seeing signal of change, even with currently available measures, ABC, SRS. There is an effort that needs to be pursued in developing clinically more patient-centric endpoints, computerized endpoint, performance-based endpoint, but even with the ABC and the SRS, if you are targeting a population that is biologically defined and that you are getting target engagement, you should be able to capture behavioral change. So, rapid overview of our DPI platform, and I recall that I think 3 years ago, Sarika, Laura, our head of discovery, who was trained at Dan Gishman's lab, gave a more thorough presentation of our DPI platform. But our DPI platform is really a machine learning and systems biology driven platform that enables to stratify these highly heterogeneous populations of patients with NDDs into biologically more similar subgroups of patients with molecular alterations in these patients converging at the pathway level and resulting in the presence of observable non-behavioral clinical signs and symptoms in these patients, such as morphological traits. So, for example, and I'm very happy that Eric Porchen is here today. So, morphological features such as enlarged head circumference that are clearly indicative of involvement of multiple sets of genes and variants and convergence at the pathway level that is actually a feature that can enable to enrich for specific pathway activation sets of genes and increase biological signals. So that is one of the phenotypical features, but also other comorbidities, so increased prevalence of some types of diseases in these patients and these relatives, for example, some types of cancers or autoimmune disorders. What we're really doing here is we're capturing associations at higher rates than would be expected by chance of specific phenotypical features with strong genetic and biological underlying that will result in increased biological signal in these patients. On the other hand, the platform also enables to select and prioritize drug candidates targeting specific patient subgroups in a more precise and effective manner. So we do this through transcriptomic bridging, meaning that we leverage on RNA and gene expression profile in these patients to be able to sort of get a snapshot, a transcriptional signature of what is happening at this X point in time in these patients. Obviously, there are important elements related to SOPs in the way that you collect RNA so that you're not polluting your results with too much noise. You have to collect RNA at different time points, but what you are getting is an instant image of what type of sets of genes are upregulated and downregulated at a specific X point in time. Why do we do this? Because when you filter this signature for CNS and NDD relevant sets of genes and variants, you start getting an NDD relevant signature for these enriched patient populations and a starting point for translation, meaning that you can start bridging this with candidate drugs. And a few years, about 10 years ago, the Broad started developing a drug-induced transcriptomic signature, then the technology improved and expanded, and now today what we are doing in our own wet lab efforts is that we are screening for drugs in cells and looking at resulting transcriptional signature and looking for closest inverse matches with the enriched populations that we identify at first. So when I say that this is a problem-solving approach, we built this platform as a puzzle, saying, okay, first, our problems that we need to stratify patients. Second, we need to find a translational bridge to be able to match drugs with these patients. So it can work in both directions because we can start with a transcriptional signature induced by drugs and reverse engineer the phenotypical features that help us define patient inclusion criteria in trials and the reverse. So how does this translate in the clinic? So we ran several observational biosampling studies to be able to validate
are in silico hypothesis. These results actually result from clinical observational studies enrolling about 300 patients at the Greenwood Genetic Center in Cincinnati Children's Hospital Medical Center, and we were able to validate the association at a higher rate than would be expected by chance of specific phenotypical features with strong genetic and biological underlying, and show that these populations actually have specific biological signatures at the metabolomic, the data is not shown here, and gene expression level. More importantly and more tangibly and translationally focused, we also were able to collect LCLs from patients with ASD phenotype 1, so the enriched population, non-enriched population with a behaviorally clinical diagnosis of autism, and controls. And we were able to show that exposing these cell lines to increasing concentrations of cyclic AMP resulted in a specific response in lymphoblastinoid cell lines from phenotype 1 patients, specific metabolic response, so an ADH production, which we did not observe in non-phenotype 1 in control. So this points to specific metabolic alterations in this subpopulation. We also replicated this experiment with our drug, our lead candidate STP1, and obtained the same results. This is important because actually similar results were reported in a monogenetic population about 10, 15 years ago by Liz Berry-Kravis, who established that Fragile X patients had abnormalities in cyclic AMP in the same way, in the same type of cell line. So what we're showing here is that if you sufficiently enrich with a systems approach a patient population with autism, you can capture biological signal and you can capture pathophysiological signatures that are actually known in the monogenetic disorders with high penetrance for autism. So this is very encouraging because this is showing that we don't need to define each individual patient with autism to be able to capture biological convergence and have a starting point for drug development. So ASD phenotype 1 and STP1. So phenotype 1 characteristics, because usually the question that I get then is, how do you see any clinical convergence in ASD phenotype 1 patients? So we observe interesting facts in these patients. The first one is that they have mild to moderate intellectual disability with, there's a large confidence interval, but so basically they a mean IQ of 65. What we also do observe is that they have consistent high hyperactivity scores in the ADCC scale. And actually, when you look at the cyclic AMP data, you immediately converge back to Fragile X, because high hyperactivity is actually a key core behavioral characteristic of Fragile X. So we're not saying that biology is entirely predictive of behavior. We don't believe it is, but we believe that these are interesting features to consider in this population. Also potential impairment in processing speed. So this is interesting because our STP1 candidate is a combination, so fixed dose combination of ipidilastin and bumetanide. This is not considered drug repurposing or 505(b)(2) because when you have a novel combination and a fixed dose combination, this is considered, from a regulatory standpoint, a new chemical entity. So we have all the non-clinical data to be able to support this development, and this was done before we entered IND-enabled studies. So PDE4/3 inhibition is the main effect driver we're looking at here. Why do we also use bumetanide? Not with the same driver in terms of pathophysiology is the rationale supported by the Zappis studies, but more because when you increase cyclic AMP or chronic increase of cyclic AMP results in the activation of NKCC1. So we're seeing these loophole pathways, and this is something that we addressed in our discovery process. So processing speed is impaired in general in ASD. This is quite a consistent finding. In intervention, behavioral interventions that end up in positive change. So basically, PRT, ABA type interventions that do result in positive change in patients usually appear to first be triggered by changes in processing speed. So changes in processing speed in behavioral intervention has been shown to be predictive of medium to long-term improvement in social communication capacity, which is core symptoms of autism. So sort of this trigger mechanism, and why do we think this is important? Because this is subject to rapid change, and this can be observed on a very short time period in clinical intervention. So PDE inhibition in general has been shown in elderly populations, typically developing elderly populations, to improve processing speed and executive functioning. So this is an interesting feature to note. Now I'm going to leave Balthazar, our Chief Medical Officer, to present the trial design of the Phase 1b. Balthazar, maybe you want to quickly introduce yourself, your own background, and your own experience of failure in drug development in the NDD field before you present the design.
Okay, so basically, my name is Balthazar. In my clinical roles, my training, I've been working for more than 30 years in I have the opportunity to get into the neurodevelopmental field because the work is conducted in larger phase 2, phase 3 studies in adults, adolescents, and even younger kids now with collaboration with Lynn credits. So we have a lot of failures, and this basically led us to try to understand what happened in terms of the failures that we have had in the past. We heard from patients, we hear from family members, and even some clinicians that there were anecdotal reports that patients responded well to the therapies. So by looking into the technology, I think this is a good opportunity to test that hypothesis. And as a matter of fact, this is the first clinical study that it was being done with the STP1. I have to mention to you that due to the population selection, which we define as ASD-FED1, we have to screen a large number of patients, and these would have not been achieved with the collaboration of Quick Erickson at CCHMC, who basically screened more than 250 patients in order to identify 20 to 30% of these patients with the characteristics that Lynn mentioned that we call ASD-FED1. Once identified the patient, we basically start this study. The study was not powered to show differences, statistical differences. It was mainly, the main objective was to test safety and tolerability at lower dosages of ASD-FED1. So the fact we have two cohorts, one cohort went into a lower dose, which is 5 milligrams of milliliters, and 1 milligram of NCTT1. Once the patients participated in this cohort finished, independent safety monitoring committee assessed the safety of the molecule and allowed us to continue to the next cohort. The study was completed, as again I mentioned to you, we evaluate mainly safety and tolerability and SAEs. We want to also characterize the PK. We wanted to evaluate target engagement, and we use EEG, ERPs, and synchronization in order to evaluate these. Additionally, we have some other PD parameters like the ABCC scale, the SRS, and the NIH toolbox. All these characteristics and lessons learned we will plan to implement in the next study, which is the phase 2. In the next slide, you see on the left side, you see the concentration in nanomolar per ml of the molecules in question, the PDE4 inhibitor as well as the NKCC1. You can see that there is a very nice kinetics, and basically the kinetics are similar to the ones that has been described in the literature that correspond to these molecules, so not being surprised. On the right side, you will see the adverse events. And I apologize because of the lowercase, but I hope you can see that we have in total 9 adverse events. All of them, they were mild. They will not require any medical intervention, and all of them, they were transient. Only one of them was labeled as drug-related, and this is polyuria, which is a typical effect that we observe in patients treated with mental health. So the molecule was well and safe tolerated. And in the next slide, the next slide, what you see is basically the target engagement that we observe. In the left side, you can observe the EEG data showing the different groups, placebo, 5 milligrams, and 10 milligrams of STP1. The color-coded mentioned the increase of gamma waves. We observe that ASD ones, they do have a high increase of the gamma waves. And this increase of gamma waves, once we treated with STP1, this gamma activity was reduced, and it was reduced statistically significant at the P level of P0.06. So the STP1 induced a change in gamma waves in the whole brain, but additionally induced gamma wave changes in regions of interest that induce again a dose-dependent decrease in these areas of interest. In the right side, what you see is basically a correlation of the administration of PDE4 or Unidas in correlation with the concentration at 1 hour plus dose. And you can see that in areas of interest, we observe a very nice correlation in between the gamma waves and the concentration of the molecule. Now, in the next slide, and I apologize for this complex slide. This data come from the lab, from Ernie Papadopoulos, which is working with Craig Erickson, and he's an expert in this field. Basically, although it is relatively novel in neurophysiology research, this chirp stimulation, it has been used successfully to drive and to observe neural activity in gamma frequency in humans and as well in rodents. And more importantly, it has been demonstrated to be sensitive to pharmacological intervention. In this particular experiment, we used chirp stimuli to evaluate the neural synchronization across a wide range of gamma waves from 1 to 100 hertz. Participants heard an auditory chirp stimuli, which is generated at 1,000 hertz tone, that the amplitude modulate by a synchronous linear increase in frequency from 1 to 100 hertz in 2 seconds. In the left side, you can see the placebo group. That the red color there is basically when the subjects were able to detect the chirp. They were not able to lock this sound over time, so you don't see. You see basically the noise and signal ratio is really fine. When we compare in the STP1 at 10 milligrams, you can see a very nice increase and lock of the signal of the chirp stimuli. So the ability to synchronize high frequency neural activity, which is the signal, and to decrease the noise from the signal gamma waves with STP1 suggests an overall improvement in auditory cortex during sensory processes in this specific population of ASD1, as we described.
I just want to, I just want to further comment on this, maybe for people who are not familiar with this assay. What we're really seeing here is that the patients are locking onto the signal versus rest of the sensory stimuli. So what we're seeing and what we're hoping for is that
you will see, in terms of clinical translation, an increase in awareness, reaction time, and capacity to process external relevant signal. Sorry about that.
No, thank you.
You were being very technical, so I just wanted to also talk about clinical translation.
Apologies for that. In the next slide, I think you will see basically the ABCs score, and we have several subscales. You can see that in the left side we have the change from baseline at day 14, and day 14 is the last treatment day in the study, and we have a follow-up period of 28 days. So all the comparisons were done at day 14, and you can see that in terms of scales, they change from baseline. Negative numbers translate into improvement. Positive numbers translate into a worsening. In all the scales that I'm showing to you, you can see that there is a big decrease or a numerical increase that did not reach statistical significance, but this numerical increase was present in the SPP1 and not in the placebo. Additionally, in the right side, something that we did is basically the PK and PK modeling. And the PK and PK modeling usually help us to predict what would be the tumor dose that we wanted to have a clinically meaningful effect. In this particular modeling, you can see, which is the blue line here, is basically we can see a correlation between the ABC scores and the concentration of Ubidas, meaning that by one logarithm increase on the Cmax, we will expect to have an increase of 5.3 decrease in the ABC scores, meaning and this change is considered clinically meaningful.
You said in concentration of the Ubidas, that's concentration of STP1, no? Obviously. So SRS?
Yes, the next slide you can see the SRS scales, which is basically in the same figures as the previous one, showing again the percentage score, and you can see again going down is improvement, going up is worsening, and you can see that there are numerical differences in three different scales, particular scales: mannerism, social awareness, and motivation. And again, by doing the PK modeling, we observe the SRS that a numerical effect showing in the PK-PD predicts that by 1 unit increase on the Cmax, we will expect to have 8 points of difference in comparison to the SRS. Again, in this particular study, we did not power the study to show differences between active and placebo. So in the next study, which is in the next slide, we intend in this basically what we intend to do is to have the same clinical design as we did in the phase 1, but the difference is that we will increase the dosages, we have higher dosages, we have a longer treatment period. We power the number of the study, or the power calculations, we power based on the PK-PD modeling, meaning that we will expect to see a change statistically meaningful when we compare STP1 versus placebo. The next is your slide, so I pass it on to you.
Yes. So the previous slide was really a summary of the findings. Once again, as Balthazar mentioned, we did not power the study to reach statistical significance. This was a POC study and also a safety and tolerability study. We will be powering the study sufficiently in the phase 2 as presented by Balthazar. We will note that there is an encouraging element with regards to precision medicine and pursuing precision medicine design in neurodevelopmental disorders, is that once you get a subpopulation, actually you don't need to recruit as many patients as you would in a typical phase 2 because your signal is stronger. So we're actually looking on the basis of our PK-PD modeling to recruit
only brackets, 80 patients in this study, and that would be sufficiently powered to capture the signal. So this is encouraging because the efforts that are required to funnel in these specific subpopulations is quite comprehensive. And once you get this population, you can run smaller sample size studies and get some signal. Where Studica is today in its effort to develop a technology focused on translating precision medicine for patients with NDDs. We've been able to validate, this was not the data presented today, but we've been able to validate in two separate independent studies the construct validity of ASD phenotype 1, meaning the association at higher rates than would be expected by chance of specific phenotypical features with these strong genetic and biological underlyings being predictive of a specific biology. We also ran similar studies for a second population called ASD phenotype 2, overall in a population of 265 patients for the second STP phenotype 2 study, and about 300 patients in ASD phenotype 1. So we've been able to capture biological signal, proof construct, capture biological signal in these patients and select and translate this into select drug candidates that we are now going to be advancing for STP1 into phase 2 and for STP2 also into a phase 2 study starting mid-2023. We also have collaborations with different clinicians and academic centers, including some investigators who have participated in previously failed clinical trials with good molecules, actually, because one of the things that I was speaking about 3 days ago at the Neurodevelopmental Disorder Drug Development Summit is that actually there are plenty of valid candidates that have been advanced in the NDD field. They just are not, they're just being given to a population that doesn't exist. So if we are able to target more specific populations, there is hope that we actually do have current pharmacotherapies that can benefit specific subpopulations. And here we collaborated with investigators who had participated in previous trials, and we did some blind recalling of responder patients to these previous studies, and with one of these studies we reached 80% specificity and sensitivity in blindly recalling responders, and in a second study 60% specificity, 80% sensitivity and 60% specificity, so quite encouraging results. I think it was 90 and 60, I apologize. Okay, so any questions so far? I know that we went over time, but we were quite excited to be able to share this work and the fact that we're going forward in these two phase 2s with valid candidates for subpopulations. I also want to thank Balthazar, who managed pretty well under stress of not having any audio. Thank you, Balthazar.